Hi, my name is Mike Wong, and I'm a computational biologist here at the Center for Computing for Life Sciences at San Francisco State University. What I hope to convey in this presentation is that we live in a sea of data and that we need tools to navigate these unknown waters. Just as naval explorers used compasses to create maps of the land and seas, we can use statistical tools to orient ourselves and find important landmarks in our data. This presentation introduces three related state-of-the-art protein research studies that use PCA, principal component analysis, or variable importance techniques. While I write software for my day job, I also consider myself to be a biologist, like this distinguished gentleman, Charles Darwin. Using his theory of evolution, we can trace going backwards by following genetic differentiation events, taking us back to some of the earliest living creatures on Earth. It is thought that 3.5 billion years ago, in shallow seas with rocks like these, the first biomolecules, membranes, genes, and proteins, self-assembled into living organisms. Proteins govern nearly all processes of life: transportation and movement, processing food into energy, even working together to create more proteins. Therefore, understanding protein function is critical to medical science. Protein function depends on structure and chemistry. We can represent protein structure using high-dimensional data of atomic properties, such as charge and volume and coordinates. Recent discoveries in protein research use molecular descriptors to answer one of two questions. What's the relationship between structure and function? And what are the features of protein structure space? These questions must overcome problems of high-dimensional data. Want to find pairwise relationships? Exponential growth. Want to resample or interpolate from a model? Exponential growth. Want to cluster? High-dimensional space is different from intuitive 3D space. Distinct metrics start to fail, and therefore we get lost. In order to figure out where we can go with the data, we need to get our bearings. One can intuitively think of PCA as navigation, orienting ourselves with regards to the most dramatic peaks and valleys of the data landscape. Mathematically, one can perform PCA using eigenvector decomposition, or SVD, singular value decomposition. More on this later. In this analogy, the most important eigenvectors correspond to cardinal directions on a map. Compared to the data landscape, the map is easier to understand, there's fewer details to confuse you, and there's a one-to-one correlation between the map and the landscape. We saw earlier that there are two ways to calculate PCA, one using eigenvector decomposition and another using SVD. SVD and PCA are closely related techniques. Both calculate eigenvectors of the covariance matrix and a diagonal matrix whose values indicate importance. As such, SVD is also used for dimension reduction algorithms, such as latent semantic analysis. So, SVD can also give us a map. One more related method is multidimensional scaling. It reduces dimensionality down to 2 to 3 dimensions, but preserves distances. It's useful for finding relationships such as clusters, but not structure. Now that we have a way to navigate our data, let's look at some research. MD, molecular descriptors, are representations of structure and chemistry of a drug or a specific region in a protein. We saw an example earlier. This study uses PCA to answer two questions. One, how do 37 different MDs compare for similarity recall tasks? And two, how do different MDs compare based on their eigenvectors? They found that related MDs performed equivalently and that the best MDs had common properties. PCA shows the most important features of MD space for similarity recall. This next study is so cutting edge, it's from the future, and is to be published in 2015. Obviously, online versions are available now. Protein function is dependent on the structure of the protein cavity, which changes shape as the protein binds to its target. Shape is recorded as a vector of atom coordinates. Changes in shape are simply the difference of atom coordinate vectors. The result is a vector. PCA is applied to all the predicted changes in a shape, finding the most expansive changes and thereby revealing the most likely mechanism for binding. Only a dozen or so PCs are needed to explain 99% of the variance in cavity movement. For this study, a nonlinear supervised classifier called RF, random forest, was trained on molecular descriptors of protein functional sites. An RF model is made of many decision trees. The trees classify each sample based on random subsets of the data. Training selects the variable from the subset that most accurately labels the sample, or in other words, gives least error. The original RF implementation provides two measures of classification errors, MDG, mean decreased Gini, and MDA, mean decreased accuracy, which can be used to rank variables in order of importance. If this study were to be revisited, PCA could be used to corroborate RF variable importance. Proteins are amazing molecules that govern nearly all life processes and are the cause or cure of nearly all known diseases. Unfortunately, proteins are complex biomolecules, and describing their structure and chemistry results in large multivariate data. I hope I've shown a small sample of how protein researchers exploit PCA and related techniques. Thanks for listening.